So we're moving again now into histology for epithelial tissue. And at this point, we've seen this image before, the histology of the duodenum, the small intestine segment. And this time we're going to focus on specifically epithelial tissue. And in the case of this sample, we're actually seeing a large amount of epithelium right here at the pointer. So this puts us, of course, histology, the study of tissues. And here we are on our level of organization spectrum here, tissue level. And we have our 4 tissues. We've covered connective, and now the focus here is on epithelial tissue. So we notice here covering exposed surfaces, both internal and external, lining internal passageways, cavities, blood vessels, and producing our glandular secretions, whether it be sweat or oil, breast milk. Of course, every type of tissue is composed of its own unique specialized cells in the extracellular matrix coming together to make some tissue. And this time the focus is on epithelium. But taking a quick look back, the makeup of connective tissue, generally speaking, again, there's unique types of cells that are developed for connective tissue. There's the extracellular matrix. And in the case of connective tissue, the majority of the tissue is composed of the matrix, as we discussed previously. However, switching on now to epithelial tissue, well, again, specialized cells, extracellular matrix. But this time we're seeing, and here's the major difference, the majority of epithelial tissue is composed of the cellular component. So there's just a large connected mass of cells with very little extracellular matrix. Really the opposite of what we described for connective tissue. So we'll continue with this now. Second category of tissue, epithelium, and we'll go into the specifics of this. And here's just a great photograph now representing that general makeup of epithelium. We could highlight here individual cell, and you can see how here we could view just dozens and dozens of cells that are all bordering one another with very little room for extracellular matrix. So a large mass of cells for the most part. So starting with epithelium, we find this tissue connected right at the surface of the body. And these epithelial cells are bonded together. So we have these sheets. You can think of it like a quilt. So a broad sheet of interconnected cells that cover our exposed surfaces. But it's not only our external surface, such as our skin and hair, but also any internal surfaces. So lining the inside of body cavities, the inner surface of any ducts and pore structures. So external surfaces, and lining the border of internal hollow cavities as well. That's where we find epithelium. So there's always that connection to the surface. And traits now that we find in epithelial tissues are spelled out here. And we'll continue with these on the next few slides. But epithelial tissues have this trait of cellularity. And this goes right to this trait here that there's an interconnected sheet of cells. So we'll describe how epithelial cells are bonded to their neighbors. Also, since these tissues cover the surface, what holds them in place and connects them down to deeper structures is now what's known as a basement membrane. So this is what attaches the superficial epithelium onto the deeper surfaces of our body. Individual epithelial cells have this trait of polarity, lacking a blood supply. And with mitosis, we rapidly produce new cells to replace older ones. So we'll expand upon these traits. So this first one here, the trait of cellularity, we can make a little sketch. I'll just draw these 2 little boxes here. So these represent two neighboring epithelial cells. We're drawing the cell membrane, and their cell membranes of neighboring epithelial cells are now directly bonded together via various types of cell junctions. So this is how we get that sheet or quilt-like description for epithelial tissues. Their cell membranes are bonded together, and that's how we get this broad sheet of cells that's covering that exposed surface. It's now only on the very deepest edge of epithelial tissue is where we have that attachment structure known as a basement membrane. So if we add this to our sketch here, deep, of course, we know means away from the surface. The exposed surface is the superficial end, is one name for it, one term to describe that. And it's only towards the deepest end where we have that attachment structure known as the basement membrane. And this is what attaches the superficial epithelial tissue and bonds it down to the deeper tissues of the body. Another trait now of polarity, if we look at an individual epithelial cell, the opposite ends, and there's the term superficial versus deep, have distinct features. So we can still expand upon this sketch here. On the superficial side, we could draw in, for example, some microvilli or some cilia. Certain organelles may be found closer to the surface or deeper, and one example is already here. The basement membrane is only ever found towards the deeper end, and we're just describing that here. So the basal end, the deeper side of the cell, that's the only location where we find the basement membrane, whereas the apical or superficial, these are just synonymous terms. The apical end or the superficial end, closer to the surface, again, there's various features that are only ever found towards that side of the cell. So with one cell that has these two very distinct ends to it, the basal side with its features, the apical end with its features, that's the description of polarity. The two ends have their unique structures. So here's some much better textbook illustrations to describe that. So the first one, to trait of cellularity, we can see how the group of cells here and their cell membranes are bound together via various types of cell junctions to make this broad sheet right here at the surface. We can see here on the deeper end of this tissue, and expanding in the view here, we can see that attachment structure of the basement membrane. So that's what bonds these superficial epithelial cells down to the deeper layers of tissue. And this figure up here is much, much nicer. So again, the location of the basement membrane just to the deep end only. There's an uneven distribution of organelles. It's a good way to describe that. An uneven distribution of organelles, and especially only towards the superficial end would we find microvilli or cilia. So that's that trait of polarity. The trait of being now avascular means that there's a lack of blood vessels. So epithelial tissue does not have its own direct blood supply, but the living epithelial tissues do need oxygen, they need nourishment. So for that reason, epithelial tissue must be in close proximity to some other type of tissue which does have a blood supply. And that's usually connective tissue. And then finally, again, with the rapid pace of mitosis, we're able to now, compared to other tissues, rapidly produce new epithelial tissues to replace the older, damaged epithelial cells of our body. So we have this pretty rapid turnover, and hair is composed of epithelial tissue. So we're able to grow hair as the older tissue breaks and detaches. It's being replaced by newer tissue. So we see these traits here in all of the unique types of epithelial tissues in our body. The cells are bonded together, typically attached to the basement membrane. There is an exception we'll see much later. The cells have this trait of polarity, so opposing ends of the cells are distinct, lacks a blood supply, and again, that ability to rapidly produce new cells. So now moving forward with epithelium, when we start to name specific types, we can see that there's now different ways that epithelial cells can be layered on top of the basement membrane. So we just pointed out the deep edge of epithelial tissue has that attachment structure of a basement membrane. So if superficial to that basement membrane there's only one layer of cells, that is the description of simple epithelium. Just a single layer of epithelial cells. Each of those cells is attached onto that deep basement membrane. The term stratification truly means layered. There's multiple layers. So a minimum of 2, but of course there could be many, many more, 10, 15. But the point is, only the very deepest layer is attached to the basement membrane. And now all the other layers are stratified or layered, stacked, superficial to that. So showing that trait of cellularity, we have our basement membrane. The first layer of cells are attached to the basement membrane. And then with the trait of cellularity, cells are bonded together and they are stratified or they're stacked as they move closer to the surface. Another type of layering is called pseudostratification. And there's truly only one layer. But now due to the irregular shape of the cells, and I'll go to a photograph, some cells are longer, some cells are shorter, and that ultimately gives the appearance that they appear that the cells are stratified when they're truly just one single layer. And again, this figure here from your textbook shows. So simple layering, we want to find the basement membrane, and we just have one cell stacked superficial to that. The cells are attached laterally, that trait of cellularity, but there's no stacking. A simple layer, simple epithelium. Stratified epithelium, again, we can see the basement membrane. The deepest layer is attached to the basement membrane, and now all the subsequent layers are now simply stratified, layered on top of that towards the surface. And the photograph here showing the pseudostratification, and really what appears to be stratified, what's falsely stratified, is the appearance of the nuclei. So it does appear as if one nucleus is superficial to another, and that is the case for the nuclei. But when close inspection, every cell does contact down to the basement membrane. And here's that appearance of the pseudostratified layering. So typically when it comes to epithelial tissues, the layering, simple, stratified, pseudostratified, this is usually the first part of the name, describing how many layers there are. Another consideration now is the shape of the epithelial cell. So there's 3 basic shapes. There's what's known as a squamous shape. And the next 2 names are very descriptive, a little cube shape or a slender column shape. So cuboidal cells and columnar cells. And let's move forward with that. So the squamous shape, if we look from the deep end to the superficial edge, it's a very thin, flat cell. And when we view it, looking down onto it, there's no consistent pattern. So with that in mind, a thin cell, but a kind of a regular border shape, it always reminds me of just looking at a fried egg. You just drop an egg into a pan and just takes on some shape. but is quite thin. That is a squamous-shaped epithelial cell. A cuboidal shape, again, just like the name implies. A little boxy cube is the appearance under the microscope. So that's how it views through cross-section and gets that appearance. And we can see those here brought around into a ring. And then lastly, the slender, tall, columnar-shaped cells. So there's much more length as compared to their width. So these slender columnar cells. And again, the shape of the cell is usually the second part of the name. So going back to the concept of the layering, again, simple epithelium. That's when there's just a single layer of epithelial cells. So here on this diagram, we have, say, the deeper underlying connective tissue here. We have that area representing the basement membrane, and then just one layer of epithelial cells superficial to the basement membrane. Or a stratified example, again, if we're looking at the basement membrane, we have just that deep layer physically bonded to the basement membrane, and now multiple layers, at least two or more, that are now just attached with that trait of cellularity, superficial. So we have the stacking of the cells. And then over here, the example of the pseudostratification, where some cells grow to be longer, some cells are shorter, and it gives that appearance. And of course, I'll mention this isn't the best sketch for that, but representing the pseudostratification. The benefits for each now: a single layer, a single simple epithelium is great for diffusion. So material can diffuse through that single layer of epithelial tissue much easier. So diffusion, absorption is the benefit. Stratified layering, where multiple layers are stacked up, this is a great protective feature. So damage can be done to the superficial layers, but there's now the deeper underlying layers giving that additional protection. Whereas over here, the example of pseudostratification, commonly seen with the cilia. So that's a usual case, but not always the case. And the advantage of this pseudostratification, especially with the cilia, is any particles that are right at the surface can be swept along with the action of the cilia. So they'll move in rhythm and in concert to brush material and propel material right across the surface. And that's a good advantage to the pseudostratification, especially when it has the cilia. We have the shapes, and there's a benefit for the various shapes. Squamous, meaning that something's very thin and flat. Well, that's great for diffusion, absorption. A great example would be in the lungs, for example. The squamous epithelium in the lungs, squamous tissue that's in the capillaries. It's just a very thin barrier for diffusion and absorption. The cuboidal shaped epithelium, and you notice here, many of our body's duct structures and pores are made from a ring of cuboidal cells. And now the advantage is we could use these pores and ducts to transfer fluid through. So transporting fluids through the length of these ducts, which themselves are made of a ring of cuboidal cells. And columnar cells, such as, say, found in the intestines, that first figure at the beginning of this lecture. Columnar cells, especially rich in the stomach and intestines, this is great for absorption. Say absorption of nutrients from the lumen of the stomach and intestines and drawing that into the wall of the tissue. And then lastly here we're seeing transitional epithelium. It truly is a stratified layer. There's variation in shape, and it's especially important because it allows for distortion and stretching. And this is a rare type. We'll see it much later on when we study the urinary system. The ureters, the urinary bladder, areas that actually have this transitional epithelium, which can be distorted and stretched to change shape to accommodate for pressure. So ultimately putting all these traits together, the layering, the shape, and of course this tissue category known as epithelial tissue. This chart here, filled in for you, goes over all the possibilities. So if there's one single layer sitting on the basement membrane of the flat, thin cells, then that's known as simple, implying one layer. Squamous is the name for the flat shape. And then the ending, you could say epithelia, epithelial tissue, or epithelium. There's no difference there. So one layer of the flattened cells, simple squamous epithelia, simple cuboidal epithelia, simple columnar epithelia. All three shapes exist with the simple layering. The same could be said about the stratified layering. All three shapes are possible. And when it comes to pseudostratification, that's only ever found with the columnar shape. So pseudostratified columnar epithelium. And then of course, transition epithelium. It truly is a stratified layer, multiple layers with variation in shape and the ability to be distorted in shape. And here's just a good chart I found online showing these different examples. So squamous examples, cuboidal examples, and again, columnar examples. And I went ahead and pasted in transitional. All right, moving forward with epithelial tissue, we're now going to see the functions. So what's the activities of epithelial tissues? Up to this point, we've talked about locations and shapes. And now we're going to see now this exocrine function. So exocrine secretion, this means that some type of fluid material is being produced, and that fluid secretion is released to the surface of the epithelial tissue. So it'll be released so that it'll coat the surface, whether it's external surface, such as your sweat glands, your oil glands, or an internal surface, such as your salivary glands, releasing saliva into your mouth. And any type of glandular structure of the body is derived from epithelial cells. It's the epithelial cells themselves that are producing the secretion for that gland, and then the secretions have to be released to the surface. So in terms of the fluids produced, there's these three primary types of glands. So this first one, we've used the term before, a serous membrane. Serous membranes, such as visceral and parietal membranes, contain serous glands, the source of their fluid, mucus, and then the combination of the two is truly what's known as a mixed exocrine gland. So we'll continue there. A serous gland is producing a serous, watery secretion, so it's very fluid. A mucus secretion, just think of the mucus in your nasal cavity and your sinus cavities. Very important to your trachea and digestive organs. A mucus secretion is much more viscous, so a much thicker viscous secretion. And a mixed exocrine gland is truly one gland that produces the combination of both serous secretions from some cells, mucus secretions from other cells, and of course, it combines, it mixes, and that's what's known as a mixed exocrine gland. Our salivary glands, the production of saliva, is a great example of a mixed exocrine gland. So of course, we have these different types of fluid secretions, and all of which are now being produced by the epithelial cells of the gland. The secretion is produced internally within the epithelial cell. So you can imagine if we're looking at this gland here, this epithelial cell is producing some type of fluid secretion. Is it saliva or breast milk, whatnot? We have to now release that secretion out of this individual cell and allow it to pass into the duct. So we have three different mechanisms, three different methods of releasing that secretion out of the cell, past the cell membrane, and into the duct. And that's what these three processes here are. They all have the same result, releasing secretions from the cells, but it's their mechanism, the process differs between these three. So we'll start with merocrine. This is the most common of the three mechanisms. And we have a description of the process. So in this merocrine process of secretion, the secretions are first produced and packaged in what's called the secretory vesicles. And numerous secretory vesicles are being produced here within the cell internally. They're packaged with now droplets of that secretion. And as the arrows show, these vesicles now migrate and contact the cell membrane. And once they fuse onto the inner surface of cell membrane, that opens up a small pore, and the contents of that vesicle can now be released into the duct structure. There's a nice textbook illustration covering all three modes of secretion. And this is just a larger image of the same thing. So the merocrine process, using multiple secretory vesicles, it's just a much bigger figure here. And as that vesicle merges with the cell membrane, opening up that small window, and the contents can now flow into the pore. A second way to allow for this release of product is with the apocrine secretion. In this mode of secretion, first there's a buildup of vesicles just near the apical end of the cell, closer to the surface. And once there's a buildup of multiple vesicles, then a large portion of the cell simply detaches. It's shed off. And now all of the contents that were built up in this region is now flowing into the pores. And of course, there's little fragments of the cell itself. So this does damage a portion of the cell, but the cell can repair itself and do this process throughout its lifetime. So an example of a gland that uses this process is now mammary glands and their release of breast milk. Still a third mechanism, and again, there's a great figure in the textbook, the holocrine mechanism. The cells are now building up so much fluid secretion that in the first step, these cells are building up so much secretion that they start to swell up, and they continue to enlarge and swell up with more and more secretion to the points where the cells actually burst. So when the cell ruptures, killing the entire cell ruptures, and all of the contents of the cell are now flowing into the pores. And this is done in what's known as a sebaceous gland. It uses this rupturing holocrine mechanism. And for now, we'll just mention a sebaceous gland. This is a oil-producing gland of our skin. And the cells of that sebaceous gland are rupturing, releasing oil ultimately as you see here, right into the hair follicle, and in turn, it leads right into the surface. So we have developed 3 different mechanisms, but again, they all have the same goal of releasing the contents from the cells to get the contents into the pores. And that's where we'll finish with this first tissue category of epithelial tissues.